By analysing the content of extracellular vesicles, or EVs, it is possible to derive information about the parent cell of origin and the physiological or disease state of an individual. EVs therefore have the potential to serve as diagnostic tools, including in settings of Alzheimer's disease, and they may help shed light on underlying pathological processes. To mark World Alzheimer's Month in September 2021, we explored findings revealing possible links between EVs and Alzheimer's disease. In this video, I will highlight some of these studies. One important observation is that protein misfolding is a common thread across neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, Creutzfeldt-Jakob disease, and amyotrophic lateral sclerosis. In these diseases, distinct proteins become misfolded and deposited in specific brain regions, sometimes with distinct trajectories. Why is this protein misfolding relevant? Well, the fact that these misfolded proteins spread across brain regions suggests that these proteins move between cells in some way. And EVs can carry proteins associated with these diseases, which raises a few questions, like, does EV-mediated protein transfer contribute to the propagation of misfolded proteins? If so, to what extent is this process responsible for advancing disease? And can the EV-mediated transfer of misfolded proteins be measured to provide diagnostic information? There are many possible explanations for how EVs might contribute to the progression of Alzheimer's disease. One hypothesis stems from the idea that lysosomal proteolysis may be impaired in Alzheimer's disease. While exosomal secretion may initially serve as a beneficial mechanism used to bypass this overwhelmed degradation system, the transport of toxic proteins may be an unwanted side effect. Although there is no consensus on the underlying cause of Alzheimer's, hallmarks of the disease are well established. These include the extracellular aggregation of amyloid beta in senile plaques and hyperphosphorylated tau proteins in neurofibrillary tangles. Immunohistochemical staining of human hippocampal tissue supports a role for EVs in the pathogenesis of Alzheimer's disease. EV proteins are enriched in amyloid plaques in Alzheimer's disease as compared to a healthy control and an individual with Parkinson's disease. The same group also showed that amyloid beta cleavage occurs in a specific subset of early endosomes, and that a fraction of amyloid beta peptides are found in multivesicular bodies and released in association with EVs, at least in HeLa and neuroblastoma cells. Overall, this work sets the scene for further studies aimed at assessing the significance of the exosomal route for extracellular amyloid beta release. Another interesting line of evidence comes from studies of EVs in the cerebrospinal fluid of mouse models of Alzheimer's disease. In mice, the number of EVs in CSF increased following the intracerebroventricular administration of amyloid beta oligomers, in comparison to the central administration of a scrambled protein. This increase was largely attributed to EV contributions from the choroid plexus, which is the specialised secretory tissue responsible for CSF production. Here, Izon's QEV isolation platform was used to separate EVs from both the choroid plexus explant cultures and primary cultures. Many biomarker development programs are in the works to characterise neurodegenerative disorders based on their serologic or histological features. The Cincinnati Cohort Biomarker Program, however, is taking a different approach to disease characterisation and therapy development. The study is phenotype agnostic, which means that studies will be based on omics technologies blinded to phenotypic data. Their goal is to confirm, before the end of 2029, the first therapy that slows or stops progression of a well-defined biological type of degeneration. The analysis of EV surface proteins and EV RNA will form part of the discovery efforts following separation with Izon's QEV columns. Given EVs can shuttle a wide range of cargo to target cells and their intricate involvement with cell communication, EV-related Alzheimer's studies were inevitable, and you can read more on the topic by visiting the original article from World Alzheimer's Month 2021.